Thank you for inviting me. I know there's a very broad audience. My background is clinical, but I'm going to try and cover some of the science and any questions. We've got plenty of time for any questions anybody would like to ask. So just to introduce the Functional Gut Clinic, we established the clinic in 2013. We're the UK's leading independent provider of diagnostic testing in digestive health in the independent sector. We see about approximately 7,000 patients a year with various digestive health conditions. We're UK-wide. We do private and public sectors predominantly in a tertiary setting, which I'll explain later. We have an active clinical trials program, active R&D program, and we use state-of-the-art technology and diagnostic algorithms to try and achieve optimum patient outcomes. We also have functional gut diagnostics, which is a commercial arm of FGC, which is a joint venture set up with Commonwealth Diagnostics International, who are the breath leading diagnostics in GI in the US. That was in 2017. And in this part of the company, we do high throughput laboratory analysis, mainly of hydrogen and methane breath testing. And we do sales and distribution of test kits and equipment, and commercialization and launch of new tests from the functional gut pipeline, and launch of new products with external partners. More recently, we've set up a digestive health breath collaborative with our existing collaborations with Functional Gut, CDI and Owlstone. And the idea really now is to set the scene for what we want to do from a basic science and R&D point of view, and try and answer some of the provocative and long-standing questions in digestive health. So this is what I'm calling the orbitive perspectives of digestive health. There are lots of parties in this that are named a few here, that you've got digestive health as a whole. And orbiting around this, you have the patients, which are the most important thing. We have clinicians, scientists, we have pharmaceuticals, nutraceuticals, and all sorts of other things. But the whole field really is up in the air. And what I would describe is the perfect storm at the moment. So we have patients that are frequently dissatisfied with their diagnosis and treatments. We have clinicians who are often disinterested and undertrained. This is a low priority area for them. There's no formal training in digestive health for gastroenterologists. Scientists don't have good models to test theories. Pharmaceuticals mainly mask symptoms. They don't treat the underlying pathophysiology. Nutraceuticals are often unproven and poorly tested. The proximal colon, which is where most of the story is happening, is very hard to study, especially when it's active. There are trillions of microbes. There are thousands of active compounds to study. And there's a global pandemic at the moment, and everyone's suffering. Now, good digestive health and immunity has never been more important for people, as it treats all sorts of facets of other parts of your health, but it is very complicated. So the scale of the problem, this is a slide that I picked up a while ago, that saying that the prevalence of benign gut disorders, so irritable bowel syndrome, reflux, are increasingly common, 10 to 25% of the UK population. And if you look at the digestive health product market in the US for probiotics, prebiotics, this has reached $32.7 billion. And this is mainly through people buying these products themselves. So there's a huge problem that people are trying to treat themselves with largely unproven products. The cost of something like IBS to the NHS from primary and secondary care is over £100 million per annum. But there's often poor evidence of the efficacy of treatments in these conditions. There's poor patient outcomes. And it's generally a poor show all around. We have to do better. But realistically, nothing in the UK is going to change for the next 5 years in digestive health unless we make it happen. Having read the recent new IBS guidelines, then they're a little bit depressing. And I think we need some new exciting science to really move things forward. So the aim of the webinar today is really to consider the reality of the digestive health landscape. So let's have a realistic starting point. I want to propose some experimental models to improve our understanding of digestive health, and really to highlight the pathway to progression in digestive health outcomes. I want to generate some ideas that will form future multidisciplinary collaborations, hopefully with some of the people who are listening to this webinar today, and really to push digestive health closer to the forefront of healthcare priorities. At the moment, it's a Cinderella topic. It's sat out in tertiary care quite often. People aren't getting the help, and we need to bring it further to the forefront of people's priorities. And hopefully the end point of this will be to help generate solutions to help millions of patients that are suffering at the moment with poor quality of life. So let's face the reality. Millions of people have digestive health dysfunction. The scale of the problem is such that it can quickly overwhelm healthcare resources. And what I mean by that is that if you do have a treatment, or you do recommend that everyone should see a dietitian, then very quickly the waiting lists go through the roof. As I said before, I sit on the British Society of Gastroenterology Committee for AGIP and recently Neurogastro, but there's no formal UK training programme for clinicians in digestive health, either GPs or consultants. So you get interested consultants who follow this up as an area of interest, but there isn't a formal training programme like you would expect in liver or endoscopy, for example. The guidelines that have been produced in digestive health really focus on managing symptoms and expectations, but not really on curing the cause of these conditions. They're often described as lifelong conditions, and I'd like to challenge that a little bit. Now, access to efficacious medications is likely to be restricted in the future due to potential costs, and we've seen this with pharmaceuticals like linaclotide that are very effective in IBS-C, but it's always quite difficult to get the GPs to prescribe these because of costs. And likewise for novel diagnostics, if we develop new tests, the access to these tests are likely to be restricted due to potential costs. And yet the current resources in digestive health are really poorly targeted and often really ineffective. So lots of money is being spent, but really ineffectively. So there is money in the pot and it should be used more effectively. And access to sophisticated diagnostics and treatment really approach lies in tertiary care at the moment. And what I mean by tertiary care is you've been to your GP, you've been to your consultant, they're looking for ways to explain this problem, and then you get access to what are relatively important tests, but maybe should come much earlier in the pathway. And I think the progress in this area lies in developing new models and sharing the economic burden across the whole spectrum of healthcare from consumer all the way through to tertiary. I think moving digestive health so it mainly straddles primary care is key, because a lot of people can do a lot of things for themselves and mainly need a little bit of gentle help in primary care to get really good results. So let's move on to digestive health itself. There are 6 stages as far as I look at it, in terms of the brain, the oropharynx, getting food in there, processing this in the foregut, digesting and absorbing in the small bowel, fermenting and moving forward into the colon, and then expulsion in the rectum. And these are the 5 processes that go on there. So you ingest food, your physiology deals with that food, you digest it, you ferment it, and you evacuate it. And that's basically my life in a slide of what I'm trying to investigate on a daily basis in patients. Now, we have an existing model that utilizes quite a lot of technologies, and whether this is looking at the brain and the CNS, the HPA axis, the autonomic nervous system, or actually looking at what's happening in the mucosal level and at the microbial level. And we've got various ways we can look at this. So we can use psychological profiling with questionnaires, we can use brain imaging, we can do real-time autonomic recordings, we can look at motility, we can take biopsies, stool samples, all of these things are available. But there is definitely a missing link at the moment that's not allowing us to progress any further than we want. So where is most of the action taking place? I never gave this region much thought until about 8 or 9 years ago when I started thinking about the proximal colon, which sounds a bit strange, but you've got this area down here, right in the middle of you, that's a big bag of warm chemicals and microbes, and you've got all the waste product coming that you can't digest coming through the small bowel into this region. You've got fermentation going here. You've got a very rich blood supply and nerve supply to this region. And all of this is communicating strongly bidirectionally to the brain and back down again. So this is the area of focus for my digestive health research for the next few years. And it brings lots of other regions into play as well. So what goes on? Well, we ingest all of these different nutrients. Some of them are absorbed in the small bowel and have high bioavailability. And we have low numbers of microbes in the small intestine because we don't really need them there as such to help with digestion. As these products that are more difficult to digest get into the colon, we need a bit of help to extract energy and nutrients from that. And where the microbial density increases in the colon, and we get lots of fermentation. And this is a nice, healthy, happy gut, getting lots of varied foods going into it, and we're all living in a symbiotic state. So what are the fermentation products? So when fermentation occurs, you produce gases, and those include things like hydrogen and methane, which we commonly measure, but we also measure short-chain fatty acids. So we also produce short-chain fatty acids, that are made from bacterial fermentation and give us energy and lots of functions within the gut. So why VOCs? Bringing it around to what we want to do to move this forward. Well, VOCs are abundant in the digestive tract, but little is known about their role and effects. So VOCs can be endogenously or exogenously produced to differentiate between host and microbial functions. Exogenous production is mainly due to bacterial fermentation of ingested material. Now, previous approaches for us guys has been to utilize stool samples, mucosal biopsies, imaging, and these are all indirect measures of what we want to look at. I always think of a stool sample as like, when the party's over, you want to be in the colon when all this fermentation is happening, not at the bottom end where all the waste product is and trying to sift through it all and try and find out what went on. So what we really want to do is do some kind of real-time VOC measurement that allows for observation during in vivo functional assays. An extrapolation of VOCs to endogenous and exogenous gut functions offers a really cool investigative tool with the following advantage. It has excellent spatial and temporal resolution, which is a term I use from my brain imaging days. Spatial in the fact that we know where this is coming from, the proximal colon mainly, and temporal resolution, we can look at these things pretty much in real time as they occur, and we can track things like symptoms while they're happening in patients. We have an almost unlimited ability for sample acquisition and study repeatability within subjects, whereas taking biopsies in patients and stool samples and urine samples are really quite difficult and unpleasant. Coming in and either blowing into a machine or blowing into test tubes, much easier to do on many occasions. So we'd be able to look at variability over time much more easily. We've got incredible flexibility in terms of study design and testing the models that we produce. And it's an unrivaled safety profile. This is completely safe. No infection control risk, no tissue sampling. So it's a great, has many advantages. So how are we going to measure VOCs? We're going to hear a bit more about this at the end, but this is the amazing technology that Owlstone have set up. Here you have some adsorbent tubes that collect VOCs in real time. You have an air purification system and a laptop that controls the breathing. The subjects breathe directly into this machine, both at baseline and during stimulated states. So what's been done so far? I'm just putting this one slide up just to show how mind-bogglingly complicated this could get. This is a great study that looked at some changes between Crohn's disease patients and healthy volunteers. But as you can see, you've got feces and breath, and you've got all of these compounds, and some of them are up and some of them are down and some of them are equal. It looks a little bit overwhelming. So which approach do we take? Do we take the needle in the haystack approach, which I feel like that is looking at before, or do we look for a piece of hay in a large stack of needles, to quote Blackadder? And I think the answer is we're probably going to try and do both. So what is our piece of hay? Well, short-chain fatty acids look like a good candidate. These are produced in large numbers in the colon. And we can look at them at baseline, and we can also look at them when they've been stimulated by ingestion of carbohydrates, for example. So we also think that fatty acids are volatile, and we can pick them up in the breath. So this could be a really good starting point for us to pick up some big signals. So is it feasible? This is some really nice work from George Hannah's group at Imperial, who've been leading the way in this area, where they've been able to look at volatile fatty acids in both breath and tissue samples in patients with gastric cancer. So we think it's feasible. And there's been recent interest in using carbohydrate probes to produce both gases and VOCs through fermentation. And this is just a nice schematic showing that you can ingest various carbohydrate probes like lactose, glucose, fructose, and these can be fermented by methanogenic organisms or commensal bacteria. These will produce gases. But whilst all of this is happening, there are thousands of other chemicals that are being produced by fermentation that we're missing out on at the moment. So we know that hydrogen and methane are biomarkers of fermentation, and we can pick those up at really high levels just through breath samples into vacutainers. And we think that VOCs will also be concurrently produced in high concentrations. And this will include short-chain fatty acids as primary targets. This is our big signal, we're hoping. But we can collect everything with the breath biopsy technology. We can collect everything else, and we can look at the haystack over time to do some really cool discovery research. And this is the kind of model that we're looking at now. We're going to administer various carbohydrate probes, look at these processes and have our normal biomarkers, but then be collecting everything else to see whether there's anything novel that crops up, and we can start to build our database, our Voltalomic database. So the first thing that we've got underway is to develop a healthy volunteer model, because strangely, there's not a great deal of data that looks at the Voltalomic profile in well-characterized healthy subjects. So we take our healthy volunteers, we take them fasted, so usual preparation for a breath test. We look at baseline VOCs to get an endogenous Voltalomic profile, and then we introduce a carbohydrate probe, which is lactulose of 10 grams, and then we can measure exogenous fermentation-related VOCs at various time points. Now, in this study, we're very interested to look at the effect of oral iron, which seems to promote methanogenic bacterial activity. So these healthy volunteers will go away and take iron supplements for 28 days. So we have a well-characterized group of healthies. We introduce a single nutrient, and then we repeat the study after 28 days. We'll look at stool samples, we'll look at symptom profiles, and hopefully we should get a large signal for short-chain fatty acids to see differences between pre and post, but we'll also obtain unique discovery data set for potential target identification. So that's the starting point for us. This is underway, hopefully get this ready in the next few months. Why is it useful to use short-chain fatty acids as targets? Well, these are potential druggable targets. There are all sorts of G-coupled protein receptors and free fatty acid receptors that are in development. These receptors are in all sorts of tissue and are involved in all sorts of metabolic processes. So, if we can show that these are playing a big role in various GI digestive health states, then we have the chance to look at these in more detail from pharmacological profiling as well as from a digestive health standpoint. So there are many contentious areas in digestive health, and I'm going to give you 4 examples that we're going to go through quite quickly. So first one is proton pump inhibitor refractory GORD, which is gastroesophageal reflux disease, small intestinal bacterial overgrowth, mechanisms of irritable bowel syndrome, and digestive health in athletes. So how can short-chain fatty acids help in GORD? The important thing to realize is that the communication within the gut is bidirectional. There are certain chemical triggers that modulate gut functions both proximally and distally to the stimulus. A classic example of this is when fat in the distal small bowel triggers the ileal brake to aid fat absorption and slow down transit. However, it was about 20 years ago, there was some really nice data from a French group that showed that fermentation of carbohydrates in the colon and subsequent short-chain fatty acid production can have a similar effect. Looking into this data, we feel that this may be responsible for a different type of reflux, which is poorly treated by PPIs and may actually be promoted by their long-term use. So what this data showed was that if you ingest lactulose, this promotes fermentation, and it actually causes the stomach to relax. So what happens here is you've got lactulose ingestion, and the volume at which the stomach creates a certain tension increases here. So it goes down, but it's actually increasing, and this correlates with gas production. So there's a link to what's happening in the proximal colon and what's being triggered in the proximal stomach. And what they showed next was that if you infuse short-chain fatty acids at different concentrations, then you got a dose-dependent effect. And this was not seen with saline. So you've got fermentation going on, it has a trigger, it seems to be mediated by short-chain fatty acids. And they did a trial here looking at fructooligosaccharides given in a crossover design study. And what you've got here is the number of transient lower esophageal sphincter relaxation. So how often the bottom of the esophagus relaxed to allow gastric contents to come up. What the study showed nicely was there was a good correlation between fermentation and the number of transient lower esophageal sphincter relaxations. And if you look at here, the number of reflux episodes with the group that were on basically a FODMAP diet increased, and their esophageal exposure time here increased from 5.4 to 9.4 in the postprandial state. So you've got a very clear picture, and this is in healthy volunteers who've got intact physiology, that if you've got something going on, fermentation going on in the proximal colon, it can have an effect on the proximal gut, and this can lead to reflux symptoms that wouldn't be treated by a PPI. And in fact, PPIs may make it worse. So here you can see on the FOS diet, you've got increased heartburn, increased regurgitation, which are your common reflux symptoms, and you've got an increased number of reflux episodes. And we do expect these differences to be much bigger in GORD patients themselves. And what we've shown is that if you take patients that are not responding to PPIs, that have been referred for anti-reflux surgery, we've shown that not only do about 60% of them have evidence of gut dysbiosis, whether that's small intestinal bacterial overgrowth, methanogen overgrowth, or a combination, but they also produce much more gas to the same stimulus as the people who don't have positive reflux symptoms in that study. So what you've got there is a group of patients on long-term PPIs with dysbiosis, and if they've got dysbiosis, they're exaggerating this effect in two ways. So it's like, again, a perfect storm. And we've got other technologies to start looking at this in more detail. This is a pH impedance probe taken at night, where we're starting to see these weakly acidic events, gas events, and reflux events sitting up in the proximal esophagus. And these patients are tending to have more throat symptoms than traditional reflux symptoms. And this is something called a ResTech probe that sits in the oropharynx. And you can see again, in a few of these patients, we're starting to see these weakly acidic nighttime events that last quite a long time, that we think may be due to nighttime fermentation and gaseous reflux. This is all early days, but this is a fascinating new area to look at in GORD, which is a huge area. So how can short-chain fatty acids help in SIBO? SIBO is a toxic subject in gastroenterology. Some people believe in it, some people don't. Some people want to test for it, some people want to treat people empirically. We sit somewhere in between, but it is a real condition. It produces fermentation of ingested food in the small bowel, which makes a whole host of symptoms occur. We normally test for it with a hydrogen or methane breath test. This is a normal study showing a gas rise after 90 minutes, whereas in a SIBO patient, they start to get gas rises very early as it passes through the small bowel, and then it increases as you go to the colon, and you get an increase in symptoms at the same time in these patients. Okay, so that's the standard test that we want to do. But there are some questions about how accurately you can measure transit of the substrate, which I think are fair. So what happens here in a normal patient is that if you have easily digestible food, it all gets absorbed here. Poorly digestible food like beans, some of it gets absorbed here, but then some of it gets fermented in the colon. With small intestinal bacterial overgrowth, what you get is fermentation of even easily digestible foods in the small bowel, and that's what causes symptoms. However, the small intestine has its own unique microbiota in health. And in SIBO, this changes in distribution and number. And the patterns of fermentation should also alter depending on the species. And potentially using VOCs in SIBO may be a much more sensitive and specific biomarker of this process. And I think hope for this has come from this great work from Mark Pimentel's group, who lead at the forefront of most of this research. And this looks at microbial diversity in patients with non-SIBO and patients with SIBO. And without going into huge amounts of detail here, you can see it's a clearly different pattern. And the main thing you find in SIBO is a reduction in firmicutes and an increase in proteobacteria. So why is that important? Well, firmicutes are most abundant in healthy subjects, and an increase in proteobacteria is seen mainly in pro-inflammatory conditions. And these organisms have vastly different metabolic profiles. So by looking at things like VOCs in different regions, you should be able to differentiate the two. So you should be able to look at many differences in metabolic function. Many novel target biomarkers are possible, and it may actually help you to target antimicrobial therapy. To do this at the moment, you have to go in with an endoscope and suck out juices and do all sorts of things. If we could do this non-invasively, it would really help move the SIBO field forward. So going on to IBS, as we know, IBS is a diagnosis of exclusion, although we're being told now to consider it a positive diagnosis so patients can start on a positive treatment pathway. It's subdivided in terms of predominant bowel habit. You've got IBS with diarrhea, with constipation, with mixed bowel habit. And most treatments aim to normalize bowel habit to either slow or quicken bowel habit or bowel transit, or to reduce gut sensitivity with neuromodulatory drugs. We can use drugs that alter the microbiome, namely antibiotics, and rifaximin is the main one in use in the States, but you can do this with probiotics and prebiotics. But there's very few objective biomarkers to guide therapy. And the question is, do treatments mask symptoms or treat the underlying condition? And I think with the treatments that are being highly recommended at the moment, because there aren't any alternatives, they tend to mask the symptoms. And the real hope is in our discovery research is, can we find ways of actually treating the underlying condition and not treating IBS as a lifelong condition, and at least in a subset of patients, getting rid of IBS and getting rid of the cause of their IBS. That is the dream. So what are we going to do? We've done this data with Smart Pill, which is a capsule that measures motility and pH in the whole bowel. And what we've seen in IBS patients who have lots of symptoms, lots of cramping here, is that the pH in the proximal colon is much lower than you get in normal volunteers. And this is because you get more short-chain fatty acid production, so excessive fermentation. So this is a biomarker. And we've also shown that if you do a low FODMAP diet, which actually starves the colon of these fermentable carbohydrates, you get a change in the microbiome, and that is reflected by a reduction in pH change across the ileocecal. So the pH is higher now in patients that have a low FODMAP diet, and that seems to be correlated with symptomatic benefit because you're getting less fermentation. So this must be due to changes in the microbiome, and it does correlate with symptomatic improvement. And the group from King's, along with Chris Prober, have done a study with VOCs in stool samples to show that actually these are unnamed substances, but there were clear differences between people who responded to a low FODMAP diet and those that didn't, and those that responded to a probiotic and those that didn't. So there are features there that give us promise that actually, in addition to our existing models, adding VOCs will give us much more information about what's going on and what are the therapeutic targets for this beneficial change. So again, moving back to our model of digestion and fermentation, what we really want to do is an active comparator study in IBS-D, which is probably the most well-characterized IBS subgroup. We want to look at something that will affect the microbiome, like rifaximin, like a low FODMAP diet, and put that against the current gold standard, which is the tricyclic antidepressants for people who are not responding to treatment, and also something that's non-pharmacological, like an intestinal adsorbent. Look at baseline VOCs to look for endogenous autolomic profiles, use a carbohydrate probe to stimulate the gut, look at the exogenous profile, and then repeat this after treatment. We want to then correlate this data with symptom profile and microbiota sampling. And really, looking at the needle here, we should be able to look at a very large signal for short-chain fatty acids and see whether these play an important role. And the haystack, we will also be obtaining unique discovery VOC data for potential target identification. So this is a study we hope to start in the next 6 months. So finally, really, so how can short-chain fatty acid production help with digestive health in general and athletic performance? I think where we're moving to in the field is that prevention is better than cure. And maintenance of remission from digestive health conditions should be the goal. And what I mean by that is, instead of patients flaring, coming to us, getting treatment, what you want to do is bring that flare down, give them a condition where their gut remains healthy for much longer, and reduce their chance of flare by increasing microbial diversity or doing some interventions that actually resettle the bowel. That's our main goal now. And good digestive health is beneficial to many other facets of human health. So all of the immunological challenges we have at the moment, cardiovascular disease, obesity, all of this comes from digestive health deficiencies in some ways. So protecting the gut should be a goal for all sorts of different healthcare professions. Optimizing individual health and performance is often based on subjective factors concerning digestive health. And what we really want to do is assess individuals' baseline digestive health profiles, and that's not been possible until recently. And what we want to do is correlate these baseline profiles with normal functions, which has also not been possible, and correlate these with things like athletic performance, which has been limited. And looking at this profiling at baseline and when stimulated and during athletic activity offers real new opportunities across this sector. And these are slides that I borrowed from Neil Williams, who gave a great talk at our Digestive Health Symposium a couple of months ago, really looking at the interplay between things like athletic-induced asthma, the microbiome, and using probiotics to reduce asthma episodes, and looking at things like the microbiome, how extreme diet and performance affect permeability and gut function, and then looking at things like diet and nutrition also have these effects. So again, these bidirectional effects that ultimately affect performance. And a great study by Jamie Kew up in Liverpool showed that up to 86% of athletes suffer with GI symptoms associated with both training and competition. And in a world of marginal gains, improving performance and reducing GI symptoms and allowing athletes to operate healthily must be a huge area of improvement for a lot of people. If you're in gut pain, as we saw from Paula Ratcliffe in the famous marathon piece, then it's going to affect your running and your timings. So we really want to look at this area in great detail, and being able to measure things like breath VOCs in real time, where looking at the effects of diet, looking at the effects of fueling, should allow us to then look at some of these changes that you see between athletes and healthy volunteers. So these are microbial distributions in different types of athletes and healthy volunteers. So big differences between. So we should be able to look at differences at baseline and then see the effect of things like extreme training and performance, looking at changes that are either beneficial or deleterious, and then using the VOC assessments to identify novel biomarkers at baseline and during exertion to try and shed light onto this area. And if we can start to tailor the diet and fueling of athletes during their exertion and their rest programs, that should lead to health benefits and performance benefits. So just rounding this up, I'm hoping I'm relatively on time at around 30 minutes. What we've got at the moment is lots of different ways that we can measure gut function, whether it's transit and permeability and stool consistency, and VOCs are going to fit right in there. And then we've got all of these products, very few of which have actually gone through robust assessment. So I think by adding another layer of objectivity, this will help us to test all sorts of products and then see which ones actually do work, and put this in with things like wellness and advice apps, symptom trackers, wearables that can measure gut function as you're moving, and real-time measures. I think this model is where we're heading in digestive health across the spectrum. So in summary, we have preliminary models of ingestion, digestion, fermentation, and function. Novel breath markers obtained using breath biopsy will really increase the spectrum of these and improve the testing model. And new diagnostics that we develop from this could range from just simple Vacutainer collection kits through to sophisticated in-clinic real-time assessments. I think the products can be, that are out in the digestive health world can be tested using this model, using a combination of both objective and subjective endpoints with almost unlimited repeatability. I think this is the beauty of the study, that the tests are quite easy to perform. And treatment determination efficacy can be assessed based on mechanistic biomarkers as opposed to empirical treatment, which I don't think really helps too many people at all. And I think the outcomes studies from these studies feed back into the model. So we're continually looking to improve the sensitivity and specificity across broad areas. So concluding remarks, from my philosophical point of view, I really feel that digestive health needs to be repositioned so it straddles primary care with appropriate clinical governance pathways observed. So I think people can escalate through primary to secondary care where required, but I think the algorithms need to be relooked at. And a lot of digestive health should be, education should work on the consumer level, so people know that the products that they can buy themselves actually have beneficial effects on their gut health. I think the cost of digestive health provision needs to be repositioned and spread out across that whole spectrum of provision, from consumer all the way to tertiary, so it's much better allocated. And this will reduce the high costs incurred at a tertiary level. Very few patients should ever reach this point. And we should encourage lifestyle and proven consumer products so that individuals see the benefit of implementing these choices and help to pick up these costs. I think just as they've had to do this with things like audiology and in vision and in dentistry, I think gut health is probably next on that list. It would be great to think we could streamline access to dietetic and basic diagnostics after primary care to reduce secondary and tertiary care costs and to improve treatment efficacy by reducing empirical treatment based on new biomarker profiles. So thank you very much for your time. I hope that wasn't too much information, and I am looking forward to answering any of your questions.
Okay, thank you, Anthony, for a very interesting presentation on the diagnosis and management of digestive health and how breath analysis can play an important role today and potentially in the future through the work we'll do together using our breath biopsy technology. So my name's Huw Davies, and I'm heading up the commercial team here at Owlstone, supporting our pharma and academic customers. I'm going to give you a brief overview of our company and a more detailed description of the breath biopsy technology that Anthony touched on in his presentation. So first of all, Owlstone Medical's mission is to save 100,000 lives and 1.5 billion in healthcare costs through realizing the promise of non-invasive biomarkers on breath. We have a number of in-house test development programs for the early detection of lung damage, liver disease, lung cancer and inflammation, amongst others. We also provide biomarker services across a range of disease areas with pharma companies for applications in drug development and precision medicine, as well as academic centers in the identification and development of biomarkers for the diagnosis, prognosis, and early detection of disease. Just a bit more background on Owlstone. The company was founded as a spin-out from Cambridge University back in 2004, really to commercialize the FAMES chemical detection technology for industrial and defense applications. Owlstone Medical spun out of Owlstone Inc. in 2016 as a means to explore the emerging opportunities for the FAMES technology in biomarker detection and biological research in medicine, and particularly around breath analysis. As a company, we have over 15 years of experience in detection of volatile organic compounds, establishing the world's first high-volume breath biopsy lab. We currently have around 170 employees, with most of those people based in Cambridge, UK, where we have our headquarters and labs, with a satellite lab in North Carolina supporting our US customers. We really are at the heart of the global breath research community, including hosting the annual Breath Biopsy conference, which I'll talk about later towards the end of my presentation. So human breath contains over 1,000 low molecular weight compounds called volatile organic compounds, or VOCs, that Anthony spoke about in his presentation. So these can be from either endogenous or exogenous origin, as shown on this slide. Endogenous VOCs are produced from physiological and pathological mechanisms ongoing in the body and reflect what's actually happening phenotypically. Exogenous VOCs originate directly from the metabolism and distribution of external compounds that are introduced into the body as part of diet, environmental exposure, medication, as well as the microbiome that Anthony spoke about in his presentation. So breath analysis is an emerging but rapidly growing field, as you can see from the graphs at the top of this slide, showing the dramatic growth in both publications as well as clinical trials being run each year. You can also see the breadth of different disease areas where breath is being applied. So this does include the gastrointestinal field, with actually over 20% of clinical trials actually involving breath analysis are being run in this therapeutic area. So these are some of our customers and partners spanning top 10 pharma companies and leading academic centers like the Cleveland Clinic and Mayo Clinic, conducting project work through our breath biopsy service lab for the identification of non-invasive biomarkers to support drug development and disease phenotyping and detection. So why breath? I think Anthony talked about some of this as part of his presentation in the context of gastrointestinal health. But just to make some more general points, so of course it's completely non-invasive, pain-free and well tolerated, even by individuals that have compromised lung function. There's really an inexhaustible supply, unlike with tissues and biofluids. And I think these two reasons really mean that it's compatible with high frequency serial sampling, for example, post-treatment with therapeutics. Detection limits can be really down to parts per trillion on breath through pre-concentration from a larger volume of breath. And VOCs, as I said, are really reflective of what's actually happening phenotypically as opposed to what may happen as a consequence of your genes. It's not just for respiratory disease. As we've learned, VOCs perfuse via the blood into the airways from other tissues in the body, including the gastrointestinal system, but also the liver, heart, etc. They can be related to biological processes through chemical identification, so we can make some very clear mechanistic associations often. And also just to mention, I think it's very much an orthogonal approach to other biomarker platforms such as genomics and proteomics, where you might want to take a systems biology type of approach to biomarker work. And then finally, I think there's a real potential to translate breath analysis to point of care or even home-based testing settings in the future through sensor type technologies like Owlstone's FAMES technology that I'll talk about later. So where are we in breath omics? It's not a new field by any means. It's been around for some time. Many papers have been published in different disease areas, but inconsistencies in the methodology, particularly around the breath collection, and underpowered studies have really slowed the progress in the field. So this has resulted in really little agreement over a lot of the identified biomarkers within the same disease areas and prevented routine application of breath tests in clinics. So breath biopsy, the technology we've developed, really provides a much more consistent platform for breath collection and analysis. And Owlstone is also leading the way in terms of larger, well-designed clinical studies for breath biomarker discovery and validation. So as mentioned on the previous slide, one of the challenges in the past with breath analysis has really been around how do you collect a high quality breath sample in the clinic? So the receiver breath sampler, as you see a schematic here on this slide, is the first standardized device for non-invasive and reliable breath collection with the reproducibility required for deployment in clinical trials. So using internal fast response CO2 and pressure sensors, receiver can monitor patient breathing in real time. This allows it to track where in a patient's breath end tidal or bronchial fractions are being exhaled. The supplied software can then be used to turn on and off sampling pumps that you see here at precise time to collect the required fraction. Over a period of time, the receiver traps and stores the VOCs and pre-concentrates them onto a breath biopsy cartridge containing sorbent tubes that, again, that you can see here, which is a bit like a vacutainer for breath. These cartridges are then sent to our breath biopsy lab at ambient temperature for analysis. The receiver is currently being used in studies at over 100 clinical sites and hospitals around the world, including the world's largest breath-based clinical trial. Just to mention, we can also train up trial nurses in about an hour, so they are ready to collect samples. So it's a very straightforward way to integrate breath analysis into a clinical trial. One of the other challenges of breath analysis is dealing with the contamination that can come from the air in the environment where the breath is being sampled. The Casper portable air supply that you see here is really designed to reduce this background level of VOC contamination by supplying filtered air directly to the receiver device. So Casper's capabilities are comparable to many industrial clean air supplies, but it comes in a highly portable format, about the size of a desktop printer. And it provides a filtered air supply that greatly increases the sensitivity of the measurement while reducing the risk of false positives and false negatives in biomarker studies, as you can see from some of the figures on this slide. So if we look at the top right here, this is just showing relative VOC levels on sorbent tubes collected from ambient air and from air that's that's cleaned up using the Casper or industrial clean air supply, and you don't see much difference between these two. On the bottom left, reducing ambient contamination provides a higher signal to noise or a clean background, clearly showing the presence of an on-breath VOC compound here that would otherwise be masked by the background contamination, providing what is in effect a false negative. And on the bottom right here, we can see contamination can also increase the false discovery rate by introducing peats that are not part of the breath sample. This becomes apparent when the background is removed by Casper, as you can see here. The breath collection system also allows for a pre-concentration step to enhance the sensitivity of the method for low abundance VOC detection. It takes roughly a minute for the blood to circulate around the body. And we're typically collecting breath sample for about 10 minutes, collecting the same fraction from each time the subject breathes, capturing and pre-concentrating VOCs onto the sorbent tubes. This means that we can sample low abundance VOC biomarkers down to parts per trillion originating from the airways, as well as other organs in the body, as I mentioned previously, carried by the blood and perfusing into the airways and onto the breath. So this slide just shows our complete workflow that includes the breath biopsy collection station that's placed in the customer's lab or clinical trial site. This is comprised of the core technologies I just introduced, including the receiver breath sampler, the Casper portable air supply, and the laptop with the breath biopsy collect software that drives the receiver hardware. Once samples have been collected, the breath biopsy cartridge, including the VOCs stored on sorbent tubes, are shipped to our breath biopsy lab in Cambridge, UK for analysis. Here we perform high-resolution, accurate mass GC-MS analysis to further enhance the breadth of coverage and reliable chemical identification of VOCs, so we can associate those VOCs with the biology and past studies to build confidence around potential biomarkers. As part of our workflow that we've called our Omni assay, we have implemented a breath biopsy library, which is a growing catalogue of over 120 currently authenticated reference standards. This really allows us to provide high confidence chemical identifications, which again has been problematic in the past, of VOCs that are changing in concentration, for example, in response to drug treatment or disease. For those VOCs that are measured in a given study but don't form part of our library, we can also search against the NIST publicly available database to generate tentative IDs outside of those that are in the library. So this library-based approach really allows us to offer focused panels of disease-relevant VOCs that have been previously identified as being on breath and linked to specific aspects of the biology. This allows us then, in the same study, to provide both a hypothesis-driven approach around specific VOCs of interest, as well as open up the aperture to measure around about 600 or more VOCs in the same sample and analysis workflow with a hypothesis-free direction. We also have our evoke probe strategy. Again, Anthony touched on this in his presentation, where we can deliberately supply a high concentration of an exogenous VOC, for example, orally in a liquid form, that is subsequently metabolized by a specific enzyme in the body and excreted back on breath. This really allows us to further boost the signal to noise above background levels to probe a specific piece of biology in a way that's not always possible with endogenous metabolites. So we're deploying this evoke probe approach both in our lung cancer and liver disease test programs to probe key enzymes that are perturbed in disease states for early detection. Of course, a similar approach could also be taken in gastrointestinal disease for novel targets, or using the carbohydrate probes that Anthony spoke about as well. So here's one set of VOCs that we've identified as being on breath and related to inflammation, oxidative stress, and lipid peroxidation as one domain of biology that may be applicable for studying diseases with an inflammatory component, such as inheritable bowel disease, for example, or others, really to qualify them and validate them as non-invasive biomarkers, for example, to assess treatment response. Just to provide a bit of an overview of our services workflow, initially we would install the breath biopsy collection station with the receiver breath sampler at the clinical site. We then provide a regular supply of breath biopsy kits, which includes the ready-to-use breath biopsy cartridge that I spoke about, as well as a conditioned mask for each breath sample that's collected. The samples are collected at the clinical site along with the clinical metadata, and then the cartridges are basically sent back to our breath biopsy lab in Cambridge at ambient temperature for analysis. Here we do the GCMS analysis workflow, and then we have a team of scientists that can perform statistical analysis and interpretation of the data and provide back a report highlighting potential biomarkers. One of the other benefits of breath that's been touched on is that once VOC biomarkers have been identified and validated using our GCMS workflows, there is the potential to develop small footprint sensor type of approaches that could be used in a point of care setting or even a home-based setting. As mentioned in my introduction, Owlstone have developed a proprietary microchip chemical sensor called field asymmetric eye mobility spectroscopy, and you can see the size of the actual chip here. So this FAMES chip can be programmed in software to detect specific VOCs with high sensitivity and selectivity. And given the size of this sensor, it may be possible to integrate this technology into handheld devices for measuring specific breath biomarkers in the future. So finally, just to say, if you need more information or would like to discuss an idea around a particular study in the gastrointestinal area or other disease areas, please feel free to contact us directly at breathbiopsy@owlstone.co.uk by email. And just to mention, there's a number of other useful resources that you might like to check out. There's a introductory webinar by Dr. Rob Money from Owlstone that actually provides a number of other case studies in different disease areas. And then we've also generated this complete guide as well that can be downloaded from our website. And then finally, just to mention, I touched on this at the beginning, we have our annual Breath Biopsy conference. This year it's going to be virtual as it was last year, and it's taking place on October 12th to 13th if you're interested in that. And here's the web address there if you'd like to register your interest for that. And with that, just say thank you very much for your attention and listening.
Thank you very much, both Huw and Anthony, for your presentations. We're going to just take a few questions right now. So the first one is for you, Huw, and it's whether the breath biopsy cartridge is reusable.
Yes, it is. As part of our service workflow, we provide a regular supply of cartridges for each sample that's collected. We do also offer this technology as a product as well. So we sell the breath biopsy collection station to those groups that have the capability to do GCMS analysis in-house. And there we also supply the sorbent tubes, and there is the possibility to reuse those through reconditioning. So that is a possibility as well. Thank you.
One here for you, Anthony, is explaining the difference between the glucose and the lactulose for the test substrate for the SIBO hydrogen methane breath test. So what are the differences there?
Yep, so glucose will be absorbed quite quickly in the small bowel. So it will look at the proximal small bowel. So if the bacterial overgrowth is more distal to that, which frequently occurs when you get reflux of contents or infection down in the ileum, you wouldn't pick up SIBO. So lactulose, the problem with lactulose is that you can sometimes get fast transit of that, so it arrives in the colon too quickly. So the compromise is that we tend to use lactulose in everyone, so we get a full profile of the whole intestine. And if it looks like there's a result which is due to fast transit, usually because the peak is much higher, much quicker than we'd expect, or that symptoms aren't reproduced, then we repeat with glucose. And if anybody's got any problems with short bowel syndrome, if they've had an operation, then we'd always use glucose first as well. So we use them both, and we use our common sense and clinical judgment to get the best results.
Thank you. There's another one here for you. How do you overcome the issues with GPs being resistant to prescribe rifaximin, cost could be a barrier, was suggested.
You can't, and that is exactly why I said that the cost is the problem, because when you have clinical conditions that potentially affect millions of people, and the cost of a 2-week prescription of rifaximin is £300, then the cost could escalate quite quickly. People might want repeated treatments. So I think trying to drive costs down, come up with new treatments and everything else, but it's a real challenge for those people who want to access effective treatments. I guess the option is to pay for that treatment yourself, which isn't always very popular in an NHS setting, but people will quite happily often spend £300 on nutritional supplements and probiotic yogurts and probiotic capsules that aren't doing anything. And maybe that cost will have to be spread. But at the moment, it's a really difficult situation that the most effective treatment for a proportion of people is not readily accessible.
Thank you. There's one here for you, Huw. It's whether Owlstone has a database of VOCs.
Yes, I think touched on that in my presentation. So we do indeed have a database of VOCs. That's the breath biopsy library that I mentioned. So right now, I think it's just over 120 VOCs we have in that library, and that helps us then to confirm the identity of VOCs that we're measuring experimentally. But we are also building up a knowledge base around those VOCs as well to understand how they relate to different aspects of the disease pathophysiology and where they've been seen in other diseases in the past as well. So that's a resource that's actively being built for the future.
And there's another one here about, for you, Huw, whether VOCs and exhaled hair could be used to study the effects of a treatment.
Yeah, absolutely. A lot of the work that we do with pharmaceutical companies is looking at how VOCs change in response to active compound versus placebo. And I think, as touched on in my talk, because it's a non-invasive method, you're able to sample on a fairly regular basis with serial sampling without causing any pain or discomfort to the patient. So I think that's definitely a very clear application for the technology.
Anthony, I think this one's for you. It's how is the pH in the colon regions measured?
In the colon regions of the gut, if they're talking about where to measure it in the gut, then we've got little nasogastric tubes we can put down to various regions, or you can swallow a radio controlled capsule that sends signals out from various gut regions. So there's a couple of ways we can do it, either invasively with tubes or with little capsules that you can swallow, and then they transmit the signals to a receiver that the patient wears.
Thank you. One here for you, Huw. To what extent would diet impact a VOC profile?
Diet can certainly have an effect. It's going to affect certain VOCs. In fact, the basis of liver program, one of the biomarkers we're looking at there is a molecule called limonene, which is found in citrus fruits, but it is metabolized by enzymes in the liver. And so the level of limonene that's excreted back on breath can be an indication of liver function, actually, and that's the basis of that program. So what I would say is that a proportion of the VOCs will certainly be influenced by diet, but others will not. So it just really depends on the VOC itself. I think there's less of an issue here with breath-related VOCs than with perhaps metabolites that can be measured in blood, where diet certainly has a very significant effect.
There's a similar question here, and I think we'll make this one our last one. It's how do you know whether they're endogenous VOCs and not from exposure?
I think a lot of that comes down to the molecular identity, actually. And also we can measure these VOCs at high concentration in the ambient air as well, a lot of times as well. So I think probably the main answer to that is through identifying what they are. We can often determine whether they're exogenous or endogenous or both, for that matter.
Great. Thank you very much to everyone. I know there's a number of unanswered questions, so we'll do our best to get the answers to you. The conversation is going to carry on the Breath Biopsy community. The link is in the chat, so if you've got any questions, we'll try and answer those for you as well. And as Huw mentioned, the Breath Biopsy conference this year is digital. It's on the 12th and 13th of October, and you're welcome to register your interest on our website from now. So thank you very much for joining us. Thank you very much to Huw and Anthony for your presentations, and we hope to see you at another Breath Biopsy event soon.